Hello, everyone, and welcome to Synapse NG, the platform where we spotlight diverse voices and foster collaborations across science, innovation, policy, and society. I am your host, Dr. Akinbobola Otitoju, and today's conversation takes us into the future of cancer treatment, a future where therapies are not just powerful, but smarter, safer, and more precise. We often hear about new cancer drugs, but one critical question is how those drugs actually reach the right cells in the body without harming healthy tissues. That's where today's guest comes in. Our guest is a distinguished pharmaceutical research scientist whose work is advancing the frontier of nanomedicine and targeted drug delivery for cancer treatment. Dr. Samson Adeyemi is a senior research scientist at the WITS Advanced Drug Delivery Platform in South Africa and an honorary senior researcher and lecturer at WITS Medical School. He also serves as an adjunct professor of drug delivery at the University of Massachusetts, Lowell in the United States. His research combines pharmaceutical sciences, biomaterials, biochemistry, and bioinformatics to design nano-sized drug carriers, including exosome-based systems aimed at improving cancer immunotherapy and precision medicine while reducing side effects. Dr. Adeyemi has published over 50 scientific articles, received the National Research Foundation Innovation and Research Grant, and plays a leadership role in strengthening genomics and bioinformatics capacity across Africa through the Nigerian Bioinformatics and Genomics Network. Beyond the lab, he is deeply committed to mentorship and training the next generation of scientists. Dr. Adeyemi, it is a pleasure to have you on Synapse NG. Welcome to the show. Now, let's start from your journey. What first drew you into cancer research, and what continues to motivate your work today?
Thank you for this opportunity. I'm glad to be here with you. My initial training in biochemistry gave me a deep appreciation for how molecular dysfunction drives disease, particularly how subtle changes in protein, enzymes, and signaling pathways can profoundly initiate physiological consequences. During that period of my initial training in biochemistry, cancer stood out to me not just as a disease of uncontrolled cell growth, but as a system-level disorder, one where metabolism, gene regulation, and cellular communication all deviate from the expected course. That complexity was what first drew me in. As I progressed, I became increasingly aware of a major gap. Many effective anti-cancer agents fail not because they are biologically inactive, but because they cannot reach the tumor in a safe, targeted, and sustained manner. This realization naturally led me into advanced drug delivery and nanomedicine. Nanotechnology offered a way to translate biochemical and molecular insights into practical therapeutic solutions by improving targeting, reducing systemic toxicity, and enabling precision intervention at the cellular and subcellular level. What keeps me committed today is both scientific challenge and also clinical urgency. Cancer remains a leading cause of mortality globally, with disproportionate impacts in low and middle income settings. Many work now sits, of course, my current work now sits at the intersection of biochemistry, bioinformatics, and nanomedicine, where we can design smarter delivery systems. We can integrate them, integrate genomics and computational insight, and move towards truly personalized cancer therapy. So the possibility of converting molecular knowledge into real world impacts easily through precision medicine is what continues to motivate my work in this field.
Right? Thank you, Doc. For someone who is without a science background, how would you explain nanomedicine and advanced drug delivery and why they could transform cancer treatment?
Yes, for someone with a science background, I often explain advanced drug delivery and nanomedicine using a simple analogy. Traditional cancer treatment is a bit like watering an entire garden just to treat one sick plant. You end up affecting everything, including healthy plants. That's why many cancer treatments cause adverse effects. Advanced drug delivery and nanomedicine change that approach. Instead of flooding the whole body with a drug, we package medicine into extremely small carriers. So small, they are measured in billions of a meter and design them to travel directly to cancer cells. Think of them as smart delivery vehicles that know where to go, when to release their cargo, their payloads. This matter because cancer is just one disease. It's not just one disease, but and not every patient respond the same way. So nanomedicine allow us to deliver the right drug at the right dose to the right place while sparing healthy tissues as much as possible. That means treatment can become effective, less toxic, and more personalized. So what makes this truly transformative for me is that these tiny systems can also be combined with genetic and molecular information. So instead of a one-size-fits-all treatment, we can begin to design therapies based on how an individual's tumor behaves. In that sense, advanced drug delivery isn't just about making drugs smaller. It is about making cancer treatment smarter, safer, and more precise.
We often hear about discovering new drugs. Why is how a drug is delivered just as important as the drug itself?
A cancer drug can be extremely powerful in the laboratory, but it is not but if it's not, it cannot reach the tumor in the body safely and in the right amount, it simply won't work as intended. In that case, or in that sense, drug delivery is what turns promising molecules into a real medicine. One of my academic mentor who I admire so much, Samia Mitrigogri, is at Harvard, a professor of drug delivery in Harvard, has a statement in pharmaceutical science that we normally usually attribute to him. And what is that? I say it in quotes. If it is not safely delivered, it is not yet a drug. So many cancer drugs fail not because they don't kill cancer cells, but because they break down too quickly in the body, never reach the tumor in sufficient concentration, or they damage healthy organs along the way. This is why patients often experience serious side effects, because the drug is spreading everywhere instead of acting where it is needed most. Drug delivery addresses these problems by controlling where, when, and how a drug is released. Advanced drug delivery system can help to protect the drug as it travels through the body, guiding it towards cancer cells, and release it in a controlled way once it arrives. This improves effectiveness while reducing harm to healthy tissue. So, discovering a new drug is only half the story, actually. Without an effective delivery strategy, even the best drug may never reach its full potential, actually. So in cancer treatment especially, how a drug is delivered can be just as important, if not more important, as what the drug is.
Thank you, Doc. Thank you. Now, your work blends laboratory science with computational modeling. How does bioinformatics make cancer drug research smarter or more efficient?
Yes, my work in biomaterials, lab experiences, and computational or computer-based modeling has really helped me a lot in my research over the years. Bioinformatics and computational biology allows us to move from trial and error approach to a more informed, data-driven decision making. Cancer is incredibly complex, very complex, and no single experiment can capture all the genetic, molecular, and cellular changes happening in a tumor. So computational tools help us to integrate and interpret these layers of information into a way that will be impossible by experiment alone. So from practical standpoint, bioinformatics helps us to identify which genes, pathways, or molecular targets are most relevant in a particular cancer type or patient group. So that means that we can actually design drug delivery systems or nanomedicine that are better matched to the biology of the tumor rather than using a one-fit-all approach. So computational modeling, for instance, also makes research very faster and more cost-effective. So before we go into the lab, for instance, we can simulate drug target interactions. We can predict how nanoparticles might behave in biological microenvironments. We can also anticipate toxicity and resistance patterns. These allow us to prioritize the important or the most promising designs and avoid unnecessary experiments, time wasting, and cost. So ultimately, these tools help us to bridge the gap between molecular data and therapy. So by combining bioinformatics with biomaterials and laboratory work, we can accelerate discovery. We can reduce development time, and we can move closer to truly personalized cancer treatment that are guided by data rather than guesswork.
Thank you, Doc. So you are working with nano-sized drug carriers, including exosome-based systems. What excites you most about their potential for precision and reduced side effects?
Yes, I work on nano-sized drug carriers, particularly of most recent exosome-based systems. And one of the things that really incites me about these technologies in terms of improving precision and reduction of side effects is that nano-sized drug carriers, particularly exosome-based systems, is their ability to align with how the body already communicate at the cellular levels. Because exosomes are naturally occurring nanovesicles that cells use to exchange signals and biomolecules. So using them as a drug carriers allow us to work with biology rather than against it. So from precision standpoint, these systems can be engineered to recognize specific cell types or tumor markers, which means drugs are delivered preferentially to cancer cells, while healthy tissues are largely spared. So this level of targeting is extremely difficult to achieve with conventional chemotherapy, for instance, where the drug circulates widely through the body. So in terms of side effects, nanocarriers help in two major ways. Number one, they protect the drug and control its release, preventing high toxic concentration in healthy organs. And secondly, because they improve delivery efficiency, we can often achieve therapeutic effects at low doses. So exosome-based systems are particularly promising because they are biocompatible. They are less likely to trigger immune response reactions and capable of crossing biological barriers that normally limit drug access. For instance, the blood-brain barrier. So overall, these technologies move us closer to cancer treatments that are not only more effective, but also has far kinder patient compliance, where precision replaces collateral damage and therapy is tailored to the biology of the tumor rather than imposed on the whole body.
Thank you, Dr. Sam. Now, here's my next question. What are the biggest challenges translating nanomedicine research from the lab into treatments patients can actually access?
Yeah, there's a lot of groundbreaking science that are taking place and that has happened over the years to reach hospitals. There are a lot of challenges in translating this nanomedicine into real treatment that patients can actually access. And one of the biggest challenges in translating nanomedicine from the lab to the clinic is complexity. Now, while nano-based system can be highly sophisticated, that same complexity makes them harder to manufacture consistently. So make it very difficult to scale them up and also regulate them. So what works beautifully in a controlled laboratory setting doesn't always behave the same way in the human body or in large scale production. Another major hurdle is safety and predictability. Nanomaterials interact with biological systems in ways that are still not fully understood, particularly over long periods. So regulators and clinicians, they need strong evidence that these systems are not only effective, but also safe, reproducible, and stable across diverse patient populations. So there are also practical and economic considerations. Many advanced nanomedicines are expensive to produce and require specialized infrastructure, which can limit access, especially in low and middle income settings. So bridging that gap means designing delivery systems that are not only innovative, but also scalable. They must be avoidable and compatible with existing healthcare systems. So on a final note, translation requires strong interdisciplinary collaboration. So successful clinical adoption depends on the chemists coming on board, the biologists, the engineers, the clinicians, and regulators all working together from right from the early stage. So when these groups operate in isolation, promising technologies often stall. So overcoming these challenges will be key in ensuring that nanomedicine moves beyond existing research and becomes real, accessible cancer treatment for patients.
All right, Doc, thank you. Now, your career spans Nigeria, South Africa, and the United States. How has working across these environments shaped you as a scientist and a leader?
Yes, working across Nigeria, South Africa, and the United States has really helped me and has shaped the kind of scientist and leader that I have become and I'm becoming, because it's a journey, right? So, for instance, working across Nigeria, South Africa, and the United States has profoundly shaped both my scientific approach and perspective as a leader. Each environment exposed me to different strengths, constraints, and priorities in research and learning. To navigate those differences has also made me more adaptable, resource conscious, and of course, collaborative. So in Nigeria, for instance, I learned the value of creativity and resilience because as of course I had my undergraduate study in Nigeria, and that really helped me to develop resilience and creativity because we have very low resource, right? Could remember during our final year research project how we improvise as student, undergraduate student. So how to ask, for example, meaningful scientific questions and make progress even when resources are limited. I developed that while actually as a student back in Nigeria. So that experience trained me to focus on impacts, relevance, and problem solving rather than relying solely on technology or infrastructure. But back in South Africa, and I was there for couple of years, a number of years in South Africa, both studying and also researching and studying. Of course, I'm still also collaborating and working in South Africa by proxy. So that exposed me to more structured research ecosystems and interdisciplinary collaboration. For instance, in our lab back in the WIT Advanced Drug Delivery Platform in Johannesburg at WITS, we have pharmacists, we have biochemistry students, students with biochemistry background, we have molecular biologists, bioinformatician, we have different group of people, computer scientists in the group, very robust group. So that's particularly at the interface of academia, public health, and policy really helped me. So it reinforced the importance of translational science and the need to align research outcomes with societal and health system needs. While I was in South Africa, that really helped me. But now coming to the States, now based in the United States, I experienced highly resourced, fast-paced research environment where innovation, scale, and cutting-edge technologies are prioritized. This has sharpened my skills in advanced methodologies, for instance, computational tools and global collaboration, while also highlighting the importance of rigorous standard and reproducibility, collaborating with industry, and also working with different network of people across the different platforms. So together, these experiences have shaped me into a scientist who values inclusivity, efficiency, and real-world impacts. So as a leader, I strive to build bridges between disciplines, institutions, and regions. Working at University of Massachusetts and now collaborating with my colleagues and students at WITS in Johannesburg, and also working with experts in the industry where I consult and do research. So that high quality science is not only excellent. This is what these different experiences has shown me across discipline, institution, and regions, that high quality science is not only excellent, but also equitable and accessible.
Thank you, Doc. Now, as a mentor and educator, what skills do you think young scientists, especially in Africa, need to develop to thrive in global research?
Wow. As someone deeply involved in mentoring and capacity building, there are a few skills that I believe African scientists need more exposure in order to compete globally. Beyond strong technical knowledge, which is the foundational one, I believe young African scientists need greater exposure to a combination of computational skills, translational thinking, and scientific communication in order to compete globally. Firstly, for instance, data literacy is very essential. So skills in bioinformatics, statistics, programming, and data interpretation are no longer optional, especially in fields like genomics, drug discovery, and precision medicine. So access to these skills allows young scientists to participate meaningfully in global research, even when laboratory resources are limited. Secondly, there is need for stronger training in translational and interdisciplinary thinking. Many talented researchers are excellent at generating data, but less exposed how to how these discoveries move from bench to real world application. So understanding regulatory pathways, product development, and intellectual property, and of course, clinical relevance helps scientists frame their work for impact. So, thirdly, scientific communication and leadership skill are often underemphasized in Africa generally. So, writing clearly, presenting confidently, collaborating across cultures, and engaging with policymakers or industry partners are critical for visibility and enhance global science.
Now, my second to the last question, Dr. Sam, thank you so much for your time. For students interested in areas like nanomedicine or bioinformatics, but feeling overwhelmed, what practical steps would you recommend?
For students and early researchers interested, it's normal. That's the first thing I want to say, to feel overwhelmed. These areas are complex and rapidly evolving. The key is to take small, structured steps that build both knowledge and confidence. Do not despise the days of little beginnings, small steps. So first, you need to focus on strong foundational knowledge. In nanomedicine, that might be chemistry, biology, or material science. In bioinformatics, that might be genetics, programming, and statistics. So solid fundamentals make advanced concepts much easier, and you can easily grasp them. And some of these courses are taught in our undergraduate level. As a biochemistry student, we learned all this in our first 3 years while in the university. So second is to get hands-on experience as early as possible, even short internships, lab rotations, or online coding exercises can teach practical skill and help you to understand how theory translate into real research. In the world of internet now, all these things are free on YouTube and some of the platforms that you can get this information. Thirdly, I would say you need to leverage online resources and communities, platforms offering courses, webinars, and tutorials. Many of them are free, can accelerate learning and connecting with peers or mentors online, open doors to guidance and collaboration. So, fourthly, identify a specific niche or question that excites you, that makes you happy. So cutting edge fields are vast. Focusing on a well-defined area allows you to make tangible progress and avoid feeling lost. And that's why, for instance, my interest over the years has been in cancer. Even in cancer, more interested in the delivery aspect, not of much of drug delivery, discovery or detection of cancer, but much more in the delivery of the different kind of nanomedicine, designing nanomedicine and treatment. That is where my area of research interest has been. So finally, I will say seek mentorship and networking actively. Early guidance from experienced researchers can help you to navigate choices and avoid common pitfalls. Identify opportunities for training and collaboration. So the most important mindset for me is curiosity. which they say is the mother of invention, coupled with persistence. So progress in these fields come incrementally, and consistent effort combined with right guidance can turn initial overwhelming situation into confidence and meaningful contribution.
Okay, thank you, Doc. And my last question for today would be looking ahead. How do you hope your work and mentorship will shape the future of personalized cancer treatment?
Yes, my vision for the future of cancer care is where treatments are truly personalized, precise, and accessible, where every patient receives therapy tailored to the molecular and cellular characteristics of their tumor rather than a one-size-fits-all approach. Through my work in nanomedicine, advanced drug delivery, and bioinformatics, I hope to contribute to platforms that deliver the right drug in the right dose and to the right targets while minimizing side effects. Integrating computational tools, genomic insight, and smart delivery systems allow me to move towards this goal more efficiently than ever before. So equally of importance is the training of the next generation African scientists. I hope that the students and early career researchers I mentor, I supervise, will not only adopt cutting-edge technologies, but also think holistically about translating discoveries into real-world impacts, designing therapies that are clinically viable, cost-effective, and ethically responsible. And that's why I belong to some, presently the vice president of the Nigerian Bioinformatics and Genomic Communication Network, the vice president communication and operations. I'm also applied to be part of the consortium of genomics students and young researchers in Africa. And of course, I'm a member of the Control Release Society of South Africa chapter and Africa Science Network, to mention but a few. So all this platform allows me to contribute to the development of upcoming scientists. So ultimately, I envision a future where the combination of innovative science, cross-disciplinary collaboration, and data-driven approaches makes cancer treatment smarter, faster, and more equitable. This goal is not just to develop advanced therapies, but to ensure they reach patients who need them most, transforming cancer from a devastating diagnosis into a manageable, even curable disease in diverse healthcare settings.
Dr. Sam Adeyemi, thank you for sharing your journey, your science, and your vision.
Thank you so much.
Today's conversation reminds us that the future of medicine isn't just about discovering new drugs. It's about discovering smarter ways to deliver them with greater precision and compassion. To our viewers, especially students and early career researchers, groundbreaking science is not out of reach. With curiosity, resilience, and the right mentorship, you can be part of the discoveries that change lives. Thank you for watching Synapse NG today. Don't forget to follow, share, and stay connected as we continue spotlighting voices shaping the future of science and society. I am Dr. Akinbobola Otitoju. Until next time, keep thinking, keep questioning, and keep connecting. Bye.